All right, hi everybody.
Hello.
I hope that you are all doing well. Remember that you have a T-cell development problem set due on Friday. Today I have a few last things to say about T-cell development, and then I'm going to move into T-cell trafficking, which hopefully will go well, though immediately before coming here, I checked one of the links that I have in one of the slides and it didn't work, and so now I'm very sad. Also, I am like a teensy bit ahead of where I was last year, aka I'm not as behind as I was last year. And so for whatever reason, I decided that meant I should put all of the T-cell trafficking slides in this lecture, even though like mathematically that's not happening. I'm still gonna have to do some of them next time too. It just made sense last night. I don't know why. So like I said, we're going to think about T-cell development and trafficking. So we left off last time talking about the Goldilocks model of thymic selection. Remember that this is happening in our double positive T cells, which are our pre-T cells. So these cells basically are getting tested to see if they get a signal in the thymus. If they get a low or no signal, then those cells die by neglect. Basically, they're getting absolutely no signal, so they must not be even binding MHC. If they get a very high signal, they will be negatively selected. So they will be actively told to undergo apoptosis and deleted. And only those that have kind of a lower intermediate signal will be positively selected, get a signal in the thymus, and survive. And I did mention that there was this fourth outcome that we will get to a little bit later. And so we talked about the fact that one kind of scary part to all of this is, of course, that every T cell that comes out of your thymus has to be able to get a low signal from self-MHC plus peptide, because that's how it could get a signal to get positively selected. So every T cell in your entire body is slightly autoreactive. And so that, is definitely a thing that is of concern. Yay peripheral tolerance, helping us deal with that, which I believe we see some of the details of that peripheral tolerance on Friday. If not on Friday, on Monday. I can't remember exactly where things fall. Also remember that when we were talking, at least in the case of MHC class 1, an MHC class 1 presentation, I told you that self-peptides are being presented on MHC class 1 by all cells all the time. So it's not that the self-peptides isn't presented. Self-peptides being presented 24/7, especially on class 1. It's not that there are no T cells that can react at all with self that leave the thymus. They all have to be able to react with self a little. It's really going to be some of these other things that we're going to see with peripheral tolerance that are going to be helping to make sure we don't have responses to self. And if you recall, this process of positive selection, this process where our developing T cell must be able to get some signal in the thymus in order to tell that cell, yes, you can live. Here's your pat on the head. That is how it ends up that T cells are restricted by the MHC of the thymus. So basically, the T cells that sort of start the process of T cell development are able to bind to all sorts of things, but only the ones that interact with the thymus structural cells, the stroma, the epithelial cells, I've used all three of those, will get a signal, and so that's how those cells will learn what self is. So here you can see if I took bone marrow from an orange mouse, put it into a yellow thymus, those cells are going to be interacting with yellow structural cells of the thymus. yellow thymic stroma, yellow thymic epithelial cells. And so when they leave the thymus, they are going to say, aha, yellow is self, because they will have only been able to be positively selected if they could bind to the yellow MHC, since that was what was available in the thymus. Or if we had the orange bone marrow going into a blue mouse with a blue thymus, those T cells are then going to learn that blue is self, because they had to get positively selected and get a signal by interacting with that blue thymus. Many of these steps where our cell is potentially getting selected will be steps now that we're starting to see happening in the medulla. This image says that the cell chooses if it's CD4 or CD8 before going into the medulla. This is literally the first time I've ever noticed that about this image. And I would say that that is not my understanding of things. So I always love when the first time you notice it is right in front of class. But the key thing I want you to see is that a lot of these selection steps are happening in the medulla. And so we're really sort of now largely in the medulla at this point. And you can also see this here, where we've got our double positive cell kind of at the end of its time in the cortex moving into the medulla. By the time that it's ready to leave the thymus, it will have gotten to being a single positive, but we haven't quite got there yet. And so these double positive cells are the mature T cells. They have an alpha chain and a beta chain. They're getting sort of an appropriate signal, not too high, not too low, just right, just like with every immunologist's favorite story, Goldilocks. And so now these are going to be our mature T cells. The one final thing that has to happen in our mature T cell is that now that T cell does need to decide if it's going to be a CD4 or a CD8 T cell. Because at first that cell was a double positive. It had both CD4 and CD8. And now it needs to only have CD4 or only have CD8 before it is able to leave the thymus to go out to the periphery. There was a lot of debate in the field about how this happened for a very long time. There were a variety of models for what might be going on, and people were really uncertain. And those are shown, some of the different models are shown on the left. On the right is a podcast interview that we did with a scientist whose lab came up with a super clever experiment to try to figure this out. And they really sort of were able to kind of nail down this model where depending on kind of different types of signaling from the MHC, the cell is able to determine whether it should have CD4 or CD8 upregulated. And so the cell is actually kind of getting some information about, am I binding to MHC class 1 or am I binding to MHC class 2? Should I thus have CD4 or should I have CD8? Doing that. But that is something that really has only been described in the past few years. And honestly, I remember reading this paper and interviewing the author, and the entire time I spent looking at it and being like, wow, this clearly answers the question, and I have no idea how I ever would have imagined doing this experiment. Like, how did you think of this? It is so like out of the box and clever, but it did answer the question. So our cell does seem to get some information on whether CD4 or CD8 is actually best in going with its receptor. And then that cell will choose to be a CD4 or a CD8. And now that cell is going to be able to leave the thymus to go to the periphery. And so we first, of course, had that cell go from the bone marrow into the thymus to undergo T cell development. Once that cell has finished T cell development, that cell is going to leave the thymus and go to some kind of secondary lymphoid organ or the periphery in order to actually make some kind of response. Very often, when we are thinking about measuring T cell development, we will often look at what kind of results we see in the periphery. And so sometimes we will look and see what T cells make it to the periphery to ask questions about T cell development. And so the next few slides are kind of different sort of experimental situations and what you see in the periphery to kind of help us understand some details about T cell development. So first of all, this is just one view of looking at T cells in the periphery. First, notice that in this image, we've got CD4 and CD8 on the axes. And you can see that in one plot we have the lymph nodes and one plot we have the spleen. I said them in different orders and I circled them, but that's okay. Those are the key secondary lymphoid organs that most immunologists think about. Those are sort of the organs that we are going to talk about as largely being the periphery, those organs and the blood. If I ask you about, drawing T cells in the lymph nodes or the spleen, if I ask either of them, I mean periphery. And I really think about lymph nodes and spleen as being pretty much interchangeable for us. And so if you see lymph node or you see spleen, that should set off the alarm bell in your head of, oh, we're doing periphery here. And what I want you to notice is that we can see in these plots, in both our lymph node and our spleen, some double negative cells, some CD4, CD8 minus cells, and we can see some single positive cells, but we don't really see a ton of double positives. We see a very small number of double positives. To be honest with you, when I think about a plot like this and when I make a plot like this, usually you shouldn't see any. It's actually a little weird to me that they saw some, but this is real data, so we'll go with it. And so if I were to draw the plot that you see here, we'll say it's a lymph node plot, and we'll have CD8 on this axis. We'll have CD4 on this axis. I'll put lymph node down here a little bit. That's kind of what I'm thinking that I'm seeing in my lymph node. And if you recall, when we looked at the thymus, we saw something that looks more like this. So in the thymus, the majority of the cells are those CD4, CD8 double positive cells, while we don't see them at all in the periphery. Those cells only exist during T cell development. And so you can see this sort of view both in my drawing version and in this plot here. There is one other thing, however, that I want to mention about these plots, also the plot that I have drawn here. If you look at these plots, we'll say of the lymph node, you can see that it lists 53.8% of the cells as being neither CD4 nor CD8. So the question is, what are those cells? That 53.8 or the 70.5% that are CD4 minus, CD8 minus in the spleen. What are those cells that are double negative cells in these two organs? Yep, Paige. Huh? They may have come from blood, but all of these could be talked about as blood cells. So what specifically kind of cells are those? Yeah, David.
Non-T cells, regular.
Most likely, those are the non-T cells. Those are the B cells. Those are the macrophages. Those are the NK cells. Those are all the other cells of the immune system that could be found in these secondary lymphoid organs. What you're looking at in these plots is a plot of all the cells in the organ. I could also, if I wanted to, do some gating before I showed you this plot. So I could do some gating, which means I could narrow down the cells I was looking at. I could make it so I wasn't looking at every cell in the spleen or every cell in the lymph node. And oftentimes what I might do is I might gate on CD3. CD3, if you remember, is on all T cells. It's part of the T cell receptor signal transduction complex. It's required on T cells. And so if I gate on CD3, I will get a slightly different plot. If I gate on CD3, I will hopefully draw on the correct screen. If I gate on CD3, I will only see the CD8 single positives and the CD4 single positives. I will not see that population of double negative cells because those are the non-T cells. And when I'm gating on CD3, I'm only looking at the T cells. I have excluded the non-T cells. And so one of the other things you're going to want to look at, again, if you're looking at, say, old exams or any of the old practice problems that are on Moodle, is that when I'm talking about the periphery and when I'm talking about T cells in the periphery, we always have to specify, did we gate on CD3? So we're only looking at T cells, or did we not gate, which means we're looking at everybody? I don't usually talk about the gating in the thymus, and sometimes students get really confused about that. Everything in the thymus is T cells. There aren't B cells hanging out in the thymus. So I don't have to gate on the thymus. So in fact, the thymus looks the same gated and not gated. But if we're actually looking at our peripheral organs, we do need to be aware of that. So one of the things that we can do is we can look at mice that have genetic differences, and we can determine what kinds of T cells end up being present in the periphery of those mice after genetic changes, and that can tell us some things about T cell development. So normal mice, listed here as control mice, if we look in their thymus, we see all four populations. And when we look in their periphery, we see some CD4 positive T cells and some CD8 positive T cells. Hooray. If, however, we have a mouse that is class 1 deficient, so it genetically does not make MHC class 1, you can see that we see double negatives, we see double positives, we see CD4 positive single positives, but we don't see the CD8 single positives. And if we look in the periphery, we only see CD4 positive cells and no CD8s. And so this tells us that we do need class 1, an interaction with class 1, in order to get CD8 positive T cells. We've got to have that interaction with the MHC in the thymus. We can also see, that also helps us know that the double negatives and the double positives came first. These types of experiments were one of the ways people were able to figure out that, double positive came before single positive, because here you had double positives, but you don't have single positives. If we have mice who are deficient in MHC class 2, then we see the other 3 types of cells, but we don't see the CD4 single positives. And when we look in the periphery, we also don't see CD4 single positives. So again, we know that in order to have development of CD4 positive cells, they must be able to interact with class 2 in the thymus. And so this sort of tells us a little bit about positive selection, and also kind of helps us understand the order of some of the events in the thymus. Another really key type of experiment that people have set up in thinking about looking at how we understand T cell development requires us to think a little bit about specific T cell responses. And so what I want you to remember here is that we have our CD4 positive T cell that responds to MHC class 2. We have our CD8 positive T cell responding to MHC class 1. They have different functions. They do all sorts of fun and exciting different things. And sometimes you might be really interested in, say, this one CD8 positive T cell. Say, perhaps you really like the CD8 positive T cell that binds to one epitope in flu. If you think back to when we talked about B cells and B cell development, I told you about some experiments that people did where they put in pre-rearranged heavy chains and light chains that gave them a B cell that responded to a particular epitope. I called those BCR transgenic mice. And what happened with BCR transgenic mice? What did we get with BCR transgenic mice? What was the outcome for the mouse? Yep, David.
Basically every mouse that had the transgenic heavy chain had that in the antibody.
Yeah, so in fact, we would often put in a heavy and a light chain. And so every single B cell in that mouse would use that heavy chain and that light chain. We would basically have a mouse where 100% of their B cells responded to one epitope, one antigen. That's really useful for us for a bunch of experimental purposes. If the mouse escapes from the mouse facility and tries to run free, that mouse is going to die because it only has an immune system that allows it to make antibodies against one thing. And oftentimes that one thing is like, hen protein or something that we like to use in the lab. So those were the BCR transgenic mice. Similarly, we can do the same kind of thing to help us understand T cells. What we generally will do in this case is we will pick a T cell that we really, 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 really like. It, for example, could be this T cell. Maybe we just love this T cell so much. And this one T cell could bind to perhaps MHC class 1, because it's a CD8 T cell. It binds to MHC class 1 presenting a particular peptide. So you can see that binding happening here. And we could take the heavy and the light chain from that T cell, and we could then insert that into a mouse, into a mouse's genome. And so again, in that case, every T cell that is going to develop in that mouse is going to basically try to grow up to become that CD8 T cell. And we're going to basically see all of our cells are going to, they're going to be double positives. And then as they do their rearrangement and they test, the only ones that are going to come out of that thymus are going to be CD8 T cells, because they all are basically growing up to be clones of this original CD8 T cell we started. If we look at in the thymus of this mouse, we'll see some double negatives, we'll see some double positives, and we'll only see CD8 positive, single positive cells. When we look in the periphery, we will only see CD8 T cells. We will have no CD4 T cells. Basically here, 100% of the T cells in this mouse will be this T cell, this CD8 T cell that potentially binds to influenza. Again, if this mouse escapes, it's in bad shape. But if you are an immunologist who would like an infinite source of this exact one T cell that you love so much, this is a really good source for it, and we can do a lot of really good experiments with it. Yep, Jazz.
So in lymph nodes and spleen, you'd only see double negatives and CD8 positives, and if you were to gate with CD3, you'd only see one big circle in the CD8 positive, CD4 negative.
Correct. People started to do sort of clever experiments using this kind of a model. And this is how we learned a lot about how T cell development works. So one thing that they did is they tried using different original T cells as the source of the heavy chain and light chain. They tried to use different sources of their pre-rearranged heavy chain and light chain. Sometimes they used a source where the receptor bound MHC class 1. Another way of saying that is that the source cell was a CD8 T cell. That's just like what you saw on the previous slide. And when they did that, the mouse only made CD8 T cells in its periphery in terms of T cells. Or sometimes they would do the same experiment using a receptor that had MHC class, that recognized MHC class 2, so basically a CD4 original T cell, and the only T cells that came out were CD4 positive T cells. And so again, that kind of emphasized the importance of those MHC T cell receptor interactions during T cell development. But they also did they did a couple other types of experiments. Here you can see they chose this T cell. They were going to make, clones of this T cell. They were going to use this T cell's heavy and light chain. And they took it out of a mouse. That's where you get the T cell from. And that mouse has a particular MHC type. In this case, the mouse is MHC type H2, is the H2K haplotype. What that means is that this particular T cell, because it is an MHC class 1 recognizing T cell, a CD8 T cell, either recognizes K of K, D of K, or L of K. I don't know which one it is. They don't have it labeled here. But the T cell doesn't recognize all three of them. It recognizes one of those with the influenza antigen. And let's imagine that it's D of K. Okay? So this T cell recognizes D of K. You put the genes for that heavy and light chain into another mouse. All of the T cells in the periphery of that mouse are going to recognize the influenza peptide on D of K. But then the trick becomes, what happens if you insert that heavy chain and light chain into a mouse where the rest of the mouse's body doesn't have D of K? What if you insert that heavy and light chain into a mouse who is of the H2B haplotype. So it has K of B, it has D of B, it has L of B. It has it from both parents, in fact. So you take the heavy and light chain from a D of K recognizing cell, and you put it into a genome where the rest of the genome is the B haplotype. If you do that, all of those T cells are gonna do the rearrangement. They're not actually gonna do any rearrangement. They're gonna make the heavy and light chain you already told them to make. Because why rearrange when you already got one? It's way easier. Yay, allelic exclusion. They're gonna all make the receptor you gave them. They're all gonna go into the thymus. They're gonna try really hard to do selection in the thymus. They're all gonna try so hard to do positive selection. They're gonna be like, here's my receptor, here's my receptor. Then what's gonna happen after that? What's gonna happen to them as they have their receptor, those T cells have their receptor that they've made, that you've given them, in this thymus? This H2B thymus. Yeah, Abigail.
They won't recognize any of the MHC molecules.
Yeah, they're not gonna recognize anything. They're gonna be in this thymus, they're gonna only have one choice of a receptor, and that one choice of the receptor is gonna recognize nothing in the thymus, because the thymus is a different MHC type. And at the end, you're gonna get no T cells in this mouse. And so this is actually one of the ways that we really started to understand the importance of that MHC in allowing for T cell development. So in fact, this is part of how we were able to figure out what's going on in T cell development. In another experiment that I don't have shown here, it's actually a really cool experiment. The problem with this experiment is that it was done early enough that we didn't understand all the parts of the immunology that were going on. And so they didn't do one of the controls they needed to do that we now know. And so now when you look at the data, it's really messy. And you're like, what's going on? I don't understand this. It's because they didn't know there's one control they needed that would have made it perfect. But one of the other kinds of experiments they did is they did something similar to this, where they had a TCR transgenic, so they had a known heavy and light chain, a known T cell. The goal was to have the mouse make only this one kind of CD8 T cell. But the antigen that they used was an antigen called HY. And the reason why HY is really cool is that HY is a protein that is encoded on the Y chromosome. And as a result, they could look at T cell development of the identical T cells in male mice and female mice. And in male mice, it was a self-antigen, and in female mice, it was a foreign antigen. And so they could actually distinguish how does T cell development differ from a self-antigen and a non-self-antigen by just looking at how the HY-specific cells developed differently between the two different mice, between the male and the female mice. And so that's also how a lot of the details of T cell development were worked out. But it's hard to look at the data from that paper because of the one missing control. So at this point, our T cells have left the thymus. They have gone into the periphery. Ta-da. And this marks an important point in our course where we now get to be in the periphery. And we now get to actually start seeing some responses to microbes because we've gotten our B cell to the periphery, we've gotten our T cell to the periphery, so now we're ready to start doing stuff in the periphery. And this is really where we're going to see, of course, our adaptive immune responses happen. So here you can see, here is that B cell developing in the bone marrow. Here are those T cells developing in the thymus. Then they both leave and head to the periphery. When we talk about them in the periphery, we can mean a few different places. One of those places is in the bloodstream, and so you can see those cells really leave the bone marrow, leave the thymus, and end up in the bloodstream. And then some of them will enter into secondary lymphoid organs. Some of them may go into barrier organs. And a lot of what we're going to talk about is sort of going between the bloodstream and in the secondary lymphoid organs. We'll talk a little bit about going to tertiary lymphoid organs or sort of other stuff. Some of the details of some of the early traffic to the barrier organs are less well understood. And so you can kind of see this here as well. When we're in the periphery, where are those lymphocytes? Well, one option is they're in the blood. And if they're in the blood, just so you know, they are able to make a full circuit of the body. So they could go from aorta back to aorta in 30 minutes. So they can basically circle around the whole body for a while. But they can also leave the blood vessels at different points to go into other organs. And so you can see they can leave the blood vessel and go into mucosal tissue, into spleen, into lymph nodes. You'll notice that the vast majority, if we add these up, are going into spleen and lymph nodes. And they're staying there for a while, 5 hours, 12 hours there. Once they leave these tissues, they can then go potentially into the lymph and then continue traveling around. And so again, here you can see this one showing 50% going to the spleen, 40% going to the lymph node. So there's a lot of interaction and movement between the bloodstream and the secondary lymphoid organ. And this is an overview of what's going on with an adaptive immune response. You're going to see this image a bunch of times over the next few class periods, where we're going to be talking about sort of some things going on with adaptive immunity. And what I want you to notice in looking at this image is that in this image from your textbook, you see this situation where we have the first infection with a particular microbe. And we see that there's this gap before which we really start to see a measurable adaptive immune response. You can see there's this period of time of almost a week before we really start to see much in the way of effector T cells or in the way of antibodies. And sometimes in the past, students have always really disliked images like this. They're like, no, I want an exact number of days, please. And to be perfectly honest with you, the exact number of days depends on the dose and the microbe and a variety of things. But this is a version I published if you want an exact number of days, though it is really specific to this microbe and this dose and things like that. So you can see in the black line, I'm looking at my normal CD8 T cells. You can see that I could actually measure them starting at day 7. I really couldn't measure much of an increase at day 4. And as I get out to 10 up to like 14, 17 is when I'm starting to kind of see the peak, and then they are going to come down after that. And so the reason why we're going to keep coming back to this over and over again is that we want to think a little bit about what the heck takes so long. Remember that VDJ recombination, the generation of a receptor that binds to that microbe, happened in development in the bone marrow or the thymus of that cell. We didn't have to wait for VDJ recombination to happen. These cells came out of the thymus ready to go. So then why did it take a week before I was able to measure a response? And so a lot of this is sort of like, what the heck takes so long in adaptive immunity? Why does adaptive immunity have this gap in terms of when we actually see this nice response? And there are lots of parts to the answer. This figure from the bottom of your textbook down here starts to show some of these parts of some of the pieces of this. And over the next few days or over the next few class periods, we're going to be seeing different things that have to happen, different things that are taken so long for that adaptive immune response. And the first big thing that we have to think about in terms of what's taking so long is transportation. Because the first thing that we have to have is we have to make sure that the B cell or T cell gets to the lymph node. We need our B cell or our T cell to make it to the lymph node. It may not start out in the lymph node, so it's got to somehow get from the blood to the lymph node. Similarly, we somehow have to get the antigen to the lymph node. So the first event that has to happen in this whole little gap of why it's taking so long to make an adaptive immune response is first, we have to get the naive B cell or T cell to the lymph node, and we got to get the antigen to the lymph node so that they can meet up. And so basically what we're talking about today is how that stuff is getting to the lymph node so that it can start to meet up. And again, you can see this is another sort of part of the what takes so long. Well, it's probably at least 24 hours to really get your antigen into the lymph node. Perhaps a similar amount of time maybe to get your T cells and B cells into the lymph node. So the first step, the first thing we got to wait for is we got to get all the stuff into the lymph node so we can start this process. So we need to talk a little bit about how the heck does the antigen get to the lymph node, and then we can start thinking about how the lymphocytes get to the lymph node. The antigen getting to the lymph node can largely happen in two ways. One of them is pretty easy. One of them is that if you recall, in our lymph vessels, we have these open-ended vessels throughout all parts of the body that are collecting up the liquid. Remember, this is what was saving us from being Jabba the Hutt way back at the beginning of the semester. And if there are random antigens, there are random microbes in the interstitial fluid, in the liquid throughout the body, they can get into the lymphatic vessel and get pushed along with the lymph into the lymph node. So we can see kind of passive movement of antigen into the lymph node pretty easily through the lymphatics. But we also have one other way that the antigen can get into the lymph node. You can also see in this image this yellow cell called a dendritic cell. And there are dendritic cells, or cells that are like dendritic cells, in many tissues. And those dendritic cells can also pick up antigens from particular areas and carry those things into the lymph node. And so we can have free antigen move to the lymph node, or we can have dendritic cells going to the lymph node. And so here you can see we could have dendritic cells, or we could have the free antigen transiting. And I mentioned a little bit about dendritic cells earlier in the semester. They are technically cells of the innate immune system. They're actually quite closely related to macrophages. But they are super important in activating adaptive immunity. So that's why we talk about them here. And so they are going to be found in a lot of different locations and potentially pick up antigen and move that antigen to the lymph node. When we think about dendritic cells, we can think about them in two ways. One of them is as what are known as immature dendritic cells, and the other is as mature dendritic cells. So the dendritic cells that are just hanging out in tissue, minding their own business, not doing anything, are immature dendritic cells. They look like this, or like this, but I actually like showing you the real, that's really what an immature dendritic cell would look like. However, our dendritic cell can get triggered to mature. And so here you can see an example comparing an immature dendritic cell to a mature dendritic cell. Really, this is supposed to be our mature dendritic cell that's gone through all of these processes, this maturation process. And so if you just look at the difference between the two, what do you see as one obvious difference between them? Yeah, Jazz.
I mean, the immature is obviously a little bit more rounded, whereas the mature has like kind of a greater surface area around it.
Yeah, so you can see this big change in surface area. You get these sort of big processes, these big pieces coming off that mature dendritic cell, and it is exactly as Jazz said, to increase surface area. This is actually why they got named dendritic cells, because the first person who saw them under a microscope thought it looked like a dendrite in a neuron because of all of the surface area and stretching outness, even though it has absolutely nothing to do with a dendrite. I say that I once got I was doing, I don't think this was NPR, I was doing a webinar during COVID for emergency room physicians, explaining how vaccines work to emergency room physicians. And I said something about the dendritic cell. And one of them was like, it does what to the dendritic cell? That means it causes brain damage. And I'm like, no, two different words, people. So yeah, there is not actually a relationship. It was just like early microscopists thought they looked the same. So you can see one big thing is that as that dendritic cell matures, it gets a lot more surface area, a lot more processes. One of the other things that that cell will start to do is it will start doing a bit more phagocytosis. It's going to be taking up a lot more stuff. But there are two other things that happen with a dendritic cell when they mature. And these are particularly important for us. One of them is that we get way more surface area, right? Why might having more surface area be important? Yeah.
To get more antigen.
What do you mean to get more antigen?
So more space to capture antigen and present them to the lymph.
So it's actually more about the second part. It's more, you have more surface area to put stuff on your surface to show off. So you can have way more surface proteins if you have more surface area. So when a dendritic cell becomes activated, you can see this example here at the bottom. It now starts to have a ton of proteins on its surface, including way more class 2, way more class 1, way more of all sorts of things that are really good at turning on T cells. I told you before that dendritic cells are the best cells for activating T cells, and it's because they have so many great surface proteins that really help T cells. So as our dendritic cell matures, it starts putting all sorts of proteins on its cell surface. I'm starting to worry about my use of this thing and whether I'm going to hit someone in the head with it. So we get all sorts of things on the surface of the dendritic cell. The one other thing that changes in the dendritic cell is that the dendritic cell suddenly moves. It was previously in the tissue, and now it wants to go to a tissue. Now, that dendritic cell that was maybe hanging out in the foot on that previous slide, now is like, I'm going to the lymph node, and is going to head to the nearest lymph node. So this cell is going to turn on lots of proteins to activate T cells. It's going to be capturing lots of antigen. It's going to move to the lymph node. It's sort of like, I think I got something. I better show this to T cells. And so this is a really, really key part of the process. You can also see this here. So you can see kind of that dendritic cells will have huge numbers of important proteins on their surface. So they have a lot of surface area, but they also have a lot of good stuff on that surface. And that is going to really help activate T cells and allow dendritic cells to be the best cells for activating T cells. And those dendritic cells are going to go to whatever lymph node is the closest lymph node to that part of the body where they have seen the microbe. And so that dendritic cell is going to go to the draining lymph node. If you remember, we have lymph nodes all over our body, each one that sort of receives the lymph from a different region of our body. Sometimes we say it drains that part of the body. And so the dendritic cell is going to head to the closest lymph node to be ready to turn on T cells with all of its great stuff that it is showing on its surface. Now, there's one last thing we have to think about with dendritic cell activation. I don't know how I skipped this slide before. See, here's my dendritic cell in the tissue. It's getting phagocytic. It's eventually going to decide to go to the lymph node so it can activate T cells. It's going to, get these nice long stretchy arms, start having all this stuff on its surface, all this great stuff to be ready to activate T cells. So this is my activated dendritic cell. You also saw activated dendritic cells here, right? Got to think of one little thing. It is mentioned both on this slide and on this slide. What makes the dendritic cell go from immature to mature? How does the dendritic cell know? Oh, I think it's time I think I got something. I think it's time to go turn on the T cells. I think it's time to go to the lymph node. I think it's time to have extra surface area. It's time to have more proteins on my surface. What tells the dendritic cell that it's time to become mature? Yep, Abigail. Yeah, it's actually PRRs. Way back to when we talked in innate immunity, we talked about how innate immune cells can use PRRs to recognize microbes. The PRR, and we talked about how that was important for cytokine production and for phagocytosis. In the case of a dendritic cell, it's also the thing that says, hey, dendritic cell, get your act together, get matured and get to the lymph node. You just got something dangerous. And so so when we have some kind of PAMP or MAMP around our dendritic cell, that's going to be the thing that's going to tell that dendritic cell to become a mature dendritic cell, to become an activated dendritic cell, and thus move to the lymph node. And so in fact, if you look back at some of the papers that you guys were looking at for the PRR assignment, some of what they were talking about there is what's going on with dendritic cell activation. So this all links back together with what we were seeing with innate immunity. Yep.
Wait, so the dendritic cells have PRRs on them and they can be stimulated with cytokines too?
Yes. But they have a lot of PRRs on and in. So you can see this dendritic cell getting stimulated by a PRR, heading into the lymph node. It's going to go to the draining lymph node. And lymph nodes are going to be the secondary lymphoid organ I mention a lot, because they're the ones I mention a lot. There's not like a particular reason. We just often talk about the lymph node. As far as I'm concerned, the spleen is just a big lymph node that, yes, has a second function too. But it's really just a big lymph node. And so I'm just going to kind of say the lymph node. It could be the spleen. And the spleen and the lymph node all have a particular structure. This is showing you the structure of a lymph node. If I was showing you a spleen, you would see the same thing. You would just see another section for its other function. But what I want you to notice is that we've got red blood, or we've got vessels, so we've got an artery and a vein, and we also have a lymphatic coming in and a lymphatic coming out. And we have different zones inside of the lymph node, one of which is often referred to as the B cell zone, also known as the follicle, one of which is also known as the T cell zone. And so it's not just that our activated dendritic cell moves into the lymph node. That activated dendritic cell is specifically going to go to the T cell zone so that it is ready to turn on T cells. And if the antigen is coming in free through the lymph, it will come into the lymphatic and could eventually end up in the T cell zone as well, though we're mostly going to think about antigen that came in on a dendritic cell here. So this, as you can see, could take a little while for the dendritic cell to phagocytose and to move and to get activated. That might take up to 24 hours. So that's part of what we're waiting for in an adaptive immune response. But then the other thing that we have to think a little bit about is, okay, well, how did the lymphocyte get there? Because the last we saw it, it left the thymus and went into the blood. So now it needs to be in the lymph node in order to meet up with this antigen and make a response. So the T cell could get into the lymph node in two ways. If the naive T cell happened to already be in the lymphatics, if it happened to have already gone in the lymph node, it could continue traveling through lymphatic vessels. So it does have that choice. But sort of the way that we're going to really talk about it is that when how that naive T cell is going to come from the blood. And so that naive T cell can also make the choice to use a blood vessel and come from the blood vessel into the lymph node because it might want to check out the lymph node. When we think about this process, we're going to see that for any white blood cell to go to any place in your whole body, there is a process, and it's the same process every time. So when we have a naive T cell and that naive T cell wants to go to the lymph node, we have this 3-4 step process. I usually think of it as 3 steps, but then I teach the 4th step, so 3 or 4 step process. We'll see this as we see the naive T cell coming into the lymph node from the blood. If we see a situation where we have an activated T cell, say you have an activated T cell that responds to influenza. It has been activated and now it wants to go to the lung to kill all the influenza it can find in the lung. It's going to go through the exact same 3-4 step process. If we have a neutrophil and we're way back at the beginning of the semester and that neutrophil is getting called to a site of inflammation, it's going to go through the same process. In fact, the book talks about it a little bit there. So we're going to be seeing this same process. In all cases, this process is called leukocyte trafficking, And so we're going to be seeing this leukocyte trafficking process both today and on Wednesday. And so you can see kind of the three examples I've just told you about here. Here we've got our neutrophil going to the site of infection. It had to leave a blood vessel and, you know, go into your hand where you slammed it on the podium or whatever. Or we might have our naive B or T cell that wants to go into the lymph node out of a blood vessel. Or we might have, you know, this activated T cell that wants to go into the lung and it's going to have to leave the blood vessel. So it's the same process in all of these cases for a white blood cell to leave the blood and go into some other type of tissue. So what I would really like to show you right now is this video. And I cannot, because when I tested it before class, I tried to get all my links up and running, and it said this video has been taken down by YouTube. And I don't know why, because I've used it for years and it's super useful. But what I want to point out, what you would see if you watched this video, you can imagine in your head this video. I think in one of the other videos you see something similar. Is that if you actually look at the blood and you watch blood cells move, you are able, you see that for their size, blood cells move really fast. So, it's one thing for you to move at 1 mile an hour, but imagine if an ant moved 1 mile an hour. It was super tiny, it was moving that amount, it was moving so many body lengths in an hour. That would be crazy fast, right? If you actually viewed it. If you look at blood cells moving, what you would easily see in this image is that a blood vessel full of blood cells moving looks like some sort of 12-lane highway at rush hour with all the cars going as fast as possible. It's slam-packed all the way across, going really, 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 really fast in the process. You can, like I said, it's really obvious to see in a video. I have a couple of things here. We actually have the ability to do microscopy on live blood vessels in a mouse, in something called intravital microscopy. So we actually can anesthetize a mouse, and we know that the popliteal lymph node is pretty close to the skin surface, and so we can actually open up the lymph node and put our microscope lens really close to it and actually watch the cells move throughout this process. Sometimes we'll put in labeled cells, though we don't always have to. But here you can see we actually can put our microscope lens right on the lymph node and watch the cells do all of these movements, both in the blood vessel and throughout the lymph nodes, through a pretty cool process. So here you can see a lymph node immobilized. We have to anesthetize the mouse and restrain the mouse in order to make this work. But it's allowed us to have some amazing images, which you will see as we talk about parts of this trafficking process. And again, if we talked about this question a little bit before about, the very big blood vessel coming out of the heart and things getting smaller and smaller. But if you were actually to look at things like physics force on those cells, you would find that the force that blood cells are encountering is more than the craziest whitewater rapids on Earth at all times. And again, I really like to think about this as a hardcore highway. Think of the craziest, many lanes, really fast driving highway that you can think of. And that's really what you should think of it looking like for these blood cells, going really fast and really packed. And that's what I would be showing you here if this video still worked. I'm so sad it didn't. This video is so good. It's all right. So let's imagine now that I am on, say, 95, driving full speed at 95. Here I am. I don't know why I'm moving my wheel so much. And I suddenly notice that, and so that you could think of that as being kind of like what the blood cell's doing. Now, the blood cell wants to leave the blood vessel and go to a tissue. That's kind of like me driving on that highway and suddenly being like, oh, I got to go to my exit. What's the first thing that there probably ought to be for me to be able to go to my exit? Like, how do I know, oh, I'm in Atlanta, Georgia? Yeah.
A sign.
So probably there's got to be some kind of sign. There's got to be some kind of marker that marks different places, right? Okay. So first of all, we're going to have to think about what exactly is going to be demarcating different locations. All right, so driving, you can pick for yourself what the max speed is. I won't tell you what speed I would go. And my exit's coming up. Should I get to where my exit is, slam on the brake, come to a complete stop, turn 90 degrees, and go over to my exit? So you're all shaking your head and making faces at me. Why not? What's wrong? Why are you making faces? Yeah, David.
If you stop completely, then you're also backing up other things that are behind you.
Okay, so if I stop completely, I'm backing up other things that are behind me. What else might happen if I stop completely? Yeah.
Just crash and die.
In David's nice world, everyone behind me stops. In a more realistic world, everyone behind me crashes into me and we all die, right? That seems bad. But I point this out because imagine what happens in your blood vessel if a white blood cell does the exact same thing. If a white blood vessel just suddenly gets to the place where it's ready to leave and comes to a complete stop and then tries to pull over, it's going to actually get sheared. Basically, the oncoming blood is going to be moving so fast by it that it's going to tear that cell in half. So it is going to crash and die. And so just like on the highway, there needs to be a little bit more process in order to allow that cell out of the blood vessel. So if I'm driving, what do I do if I want to go to my exit? What is the process? What should the process look like? Yeah, Jazz.
Well, let's say like go super fast. You'd be all in the left lane.
Okay.
Start making your way over slowly towards the exit.
Okay, so one thing I'm going to do is I'm going to get out of the middle and I'm going to move towards the side, right? So I don't want to be in the middle of the road or in the middle of the blood vessel. I need to pull over to the side. And that is actually going to be one of the first steps that we're going to see. What else are you going to do besides move over to the side? Yeah.
Then you're going to start decelerating.
And you're also going to have to start to slow down. So you really need to first slow down and move over to the side. You're not just going to be able to stay in the middle of that high-speed traffic and come to a complete stop, because that would be bad for you and everybody around you. You first have to figure out a way to pull over and to slow down. And so the first step that we see in all of these situations is that our white blood cell is going to recognize that it's at the place it wants to go. It's going to pull over and slow down, or slow down and pull over. This step is a step called, as the first step of our leukocyte trafficking process, and this is a step called rolling. So with rolling, we're going to have some protein on the surface of our vessel that marks that part of the vessel as a potential place for the white blood cell to leave. And we're going to have some other molecule on the surface of our white blood cell that is going to bind. So that interaction is going to be like the sign recognition. That interaction is also going to pull the cell out of the middle of the vessel, and that interaction is going to slow the cell down. And so all of these are going to be based on the first interaction that happens in this process, this rolling interaction. I am just going to do all of the videos next time. But this interaction that we see is mediated by something called a selectin. You've heard the word lectin earlier this semester. What was a lectin? Yeah, David. It's a protein that binds to a carbohydrate. So selectins are these proteins that are important in the rolling process, and they bind to some kind of carbohydrate ligand. So we're going to have some selectin binding to its matching carbohydrate ligand, and that's going to be the thing that starts the rolling process. And here you can see a selectin binding to a carbohydrate that's going to start this rolling process. There are really, as far as I think about it, 3 different selectins. L-selectin, P-selectin, and E-selectin, also known as CD62L, CD62P, and CD62E. They each have a particular carbohydrate that they like to bind. And so what we'll see to start the rolling process is we'll see interactions between the selectin and the carbohydrate that are sort of pulling the cell over to the side of the vessel and starting to slow that cell down. Whether or not the selectin is on the blood cell, whether it's on the leukocyte or whether it's on the vessel, varies in different situations. So L-selectin is on white blood cells, and it binds to a carbohydrate that is on blood vessels. P and E-selectin are on white blood cells, and they bind to carbohydrates that are on blood vessels. So it's a little hard for me to say the selectin's always on this partner, the carbohydrate's always on this partner, because it varies among the selectins and the carbohydrates that exist. We will continue talking about the leukocyte trafficking process and then start further talking about T cell activation on Wednesday.